familiar with the term endoplasmic reticulum, but today we want to talk about the details. So hello and welcome back to another video. Now, the ER is an organelle that is involved in creating, packaging and secreting proteins. Now, it's part of all eukaryotes, whereby in animal cells it makes up more than half of the membrane contained within one cell. The ER is a membrane system which forms flattened sacs and also tubules. It's connected to the nuclear envelope, so the outer part of the nuclear envelope can be seen as a part of the ER, or vice versa. Now, it has many interactions with other organelles, especially the Golgi, which is often the destination of the secreted proteins. Now, contrary to what textbook pictures might suggest, and as you see here, the ER is actually a highly dynamic structure, which can react to the surroundings. The distribution and the shape of the ER is regulated by integral membrane proteins, by other organelles, and also the cytoskeleton. The structure of the ER varies throughout different phases, so this can be during the cell cycle or also in development. Now, even though this is known, it's actually unclear what exactly introduces those changes. Now, let's talk about the two main types of ER, which are the smooth and the rough ER. The rough ER is characterized by ribosomes, which you see here depicted as those little dots. And they are the protein factories within our cells. Ribosomes are not static structures and they might attach and detach from the ER. And this part of the ER is the one that is close to the nucleus, and thereby it can send fast signals, which then might control protein translation rates. And one of the multiple functions of the rough ER is to synthesize lysosomal enzymes via a specialized pathway. Now, when we're talking about the smooth ER, the most striking feature is that there are no ribosomes and that we've got tubules instead of sacs. Now, actually, there are some areas which have features of both. The smooth ER takes part in synthesizing lipids like cholesterol, which is an essential component of cell membranes. And it's also crucial when we're talking about vesicles that travel from the ER to other organelles or also destinations within the cell. The smooth ER in the liver, for example, is involved in detoxing specific substances like alcohol or drugs, and the one in the brain in hormone synthesis. The sarcoplasmic ER, which is a specialized form, is involved in the calcium concentration. And this is essential when we're talking about muscle contractions. We'll hear more about that in a minute. Now, the ER is the largest organelle and comprises various functions. Therefore, it's essential for cellular homeostasis. Now, let's recap the most essential functions. So, we have protein synthesis and folding. Now, there are integral membrane proteins, but also secretory proteins, which get recruited to the ER membrane by a sequence of amino acids, and they're known as the signal recognition particle. The proteins can then either be cleaved and released into the ER lumen, or they can be released back into the cytosol where they get transported to another location. Then we have lipid synthesis, and this happens between the ER and the Golgi, where many tubules and vesicles are located. In general, those two organelles interact closely when they produce phospholipids. And here you see the molecular structure of cholesterol. Now, for calcium metabolism, we have the sarcoplasmic reticulum as indicated before, and this is the paramount storage area for calcium. There's a concentration gradient which ranges from 100 nanomolar in the cytosol to 2 millimolar extracellular, and this is essential for signaling cascades. For example, for muscle contractions, but also after fertilization. And here you see the typical scheme of signal transduction. Because, well, there's a receptor which has a signal and then it activates a second messenger, this activates another second messenger, and this results in an up or down regulation of gene expression. Well, in general, if we have cells with higher lipid synthesis or more signaling, we have more tubules, and those with higher protein synthesis have more sheets. So the shape of the ER is determined by its predominant function. I hope you enjoyed this maybe more detailed view on the ER than usually. If you did so, don't forget to give the video a thumbs up and to subscribe to the channel to see weekly uploads on scientific topics. See you in the next video.